And I'm very excited to present our last speaker for today. This is Dr. Yoel Klug from University of Oxford.
Hi everyone, I'll just get my thing up. I think it's this one. There we go. Should be able to see my screen. What's going on? Can you see, can you see my screen now? Or are you seeing just my PowerPoint slide? That's my question.
Yeah, you're good.
Okay. Because for some reason it's not. I know what's going on. There you go. Okay, great. So thank you everyone for sticking around here to this late hour, especially in Israel. And thank you for the organizers really for basically making this happen because it's been a long time coming and it's been really overdue. So I think I'd like to tell you today about the work I've been doing the past like 5, 6 years in the Cryo lab, and basically tell you about this unifying mechanism for fat storage and lipid droplet formation by protein called sapin. And I hope that you'll get the Lord of the Rings reference towards the end of the talk. Before I talk about sapin, which I'll talk a lot about today, I think I'll talk first about lipid droplets. And so lipid droplets are basically the dedicated organelle for energy storage in the cell, and they store energy in the form of neutral lipids or fat, basically, in the form mainly triglycerides. And they're also really important for energy homeostasis and for buffering lipotoxicity. You can see here on the left here some nice yeast cells with their lipid droplets here staining green, and yeast is the organism I work on. And on the right in this tomogram, you can see the lipid droplets emerging from the ER bilayer. So lipid droplets are actually formed and originate from the ER. Neutral lipids actually synthesize between the ER bilayer, and at about 5 to 10%, they can phase separate here in stage 2, where they will basically make this lens-like structure. And as they accumulate, they will bulge out into the cytosol to form mature lipid droplet. And this actually gives rise to the unique structure of lipid droplets, which I hope you can see here in this image, or basically have a monolayer versus a bilayer that every other organelle has. So they're really, really unique. And of course, this is biology, so this is way more complex than just this. And there are tons of proteins that play a role here. And today, I'd like to tell you about our stories on sapin. If you're interested in learning about some other proteins, you can look up our new publication. But sapin really is the major driver of lipid droplet formation in the cell. And in humans, if you have mutations in you will basically have a really severe form of lipodystrophy. And this is called Bernardelli-Sape congenital lipodystrophy, or BSCL type 2. And what happens with these patients is they either cannot store fat at all, or they store it ectopically, so in the abdomen or the skeletal muscle. And this comes along with a lot of really harsh pathologies. Now, sapin is conserved from yeast all the way to humans. That's lucky for it's also found in plants. And if you look at these yeast cells here, you can see healthy cells, a lot of little kind of spots there. There's the lipid droplets. If we delete sapin, we get a really striking phenotype. So basically, we get 1 or 2 supersized lipid droplets per cell. You can also get small and clustered ones. And in human cells, you usually see small and clustered lipid droplets. And this really depends on the cell type and also, of course, what the cells are eating. So basically, what media they're in and what we're feeding them. And this really brings me to my research focus, which is to ask, how does sapin actually influence lipid droplet formation? When I joined the Carvara lab, we didn't know much about sapin. We knew that it sits at this ER-LD interface. We knew that's really important for lipid droplet formation. We knew it functions as an oligomer. And we knew that it has this extended luminal domain, 2 transform domains, and some N and C termini. And we knew that if we mutate it, we get, like I showed you, this aberrant LD morphology, but also that lipid droplets, or LDs, as I'll call them a lot, are basically defective incorporating proteins and nutrilipids. So they're basically metabolically aberrant. And what I set out to do is to figure out, how does sapin actually work? And today I'll tell you about 2 stories. The first one, which is already published, mechanism of lipid droplet formation by sapin. So basically more about stages here 1 and 2. So how does actually sapin promote lipid droplet formation? And then next, I'll tell you about kind of the next frontier, some unpublished data that we're trying now to get out, which is basically about the later stages, how sapin helps in lipid droplet maturation. So to kind of get to basically a short and a long story, we got a wonderful structure here of yeast sapin, and this came along with partial structures of human and fly sapin. And I hope you can see they make this wonderful ring-like structure. They have very similar beta sandwich folds over here. In human, we have 11 subunits, in fly we have 12, and in yeast we have 10. And what was really interesting is actually this kind of central helices over here. And if we look at a side view, though, we can see that in our structure, we actually also managed to resolve the transmembrane domains. Now, I don't have time to tell you about the transmembrane domains today. So if you're interested, you can ask me afterwards. But I'd really like to focus on these central helices here. And the reason for that is that in human and fly, these helices protrude really deeply into the ER bilayer. And I hope you could see on the right here in yeast that they're really, really shallow. They basically cannot enter the bilayer. And if we look at the charge of these of sapiens, you can see that yeast is really highly charged. Basically, it's very polar. And fly and human are basically hydrophobic. Now, if you think about it, if you're going to be hydrophilic and you're going to be shallow, you're probably not going to be really good at entering the bilayer. And this was shown a bit more directly with molecular dynamic simulations. And this was done on by the labs of Stefano Vanni and Elina Ikonen. And what they showed here is this is done on the partial structure of human sapien, where they put in the structure and they modeled it with tag molecules here in brown, and they could see that those hydrophobic helices are really attracting the tag molecules. And both of these studies actually also pinpointed serine residues that through their OH, their hydroxyl moieties, actually bind the neutral lipids, bind the triglycerides, and help them basically bind sapien, and this really reduces the percentage at which they will phase separate. Now, yeast sapien is actually really bad at concentrating neutral lipids, and we showed this with molecular dynamic simulations as well. And the reason for that is, like I said, it's basically shallow and it's polar, so it can't basically do the job that the other sapiens are doing. So how does it function? And actually, yeast sapien is really, really unique. And the reason for this is that it actually requires a binding partner called LDB16, which is an ER membrane protein. And as you can see here, if we delete LDB16, we get the same phenotype we do if we delete sapien. Now, we can actually express human sapien in yeast, and this will restore normal lipid droplet formation in both single mutants, but also double mutant. And this basically suggests that in yeast, the function of sapien is broken up into two polypeptides, say one LDB16. Now, we really want to get a structure of LDB16 with sapien, and we couldn't do that, and I'm happy to discuss our failures later. But what we did do is we went to an alternative approach called in vivo site-specific photocross linking. And here, what we do is we incorporate in a natural amino acid, benzophenone, at amber stop codons. We can choose which position in the protein. We shine UV light on it, and it will cross-link to any CH bond nearby. And what we did this by putting this on sapien, and we could actually map the interactions of sapien with LDB16 and sapien to itself. And we could actually place LDB16 at the center of the sapien ring. So that was great. We now know where LDB16 is sitting, but what's actually going on here? How is LDB16 functioning? If you remember the human sapien with its hydroxyl serines, LDB16 is really packed with hydroxylated residues and this is a TR Rosetta model, so we don't have a structure of LDB16, but in the proposed luminal region, we really have a lot of hydroxyl residues. And so with this and a lot of other data I don't have time to show you today, what we think is going on is that actually sapien in yeast acts like a scaffold to position LDB16 in the right place where it can actually concentrate free-floating triglycerides and thus promote lipid droplet formation. Now, the basic idea is that all sapiens are going to work the same way. So they all form a ring. They all need hydroxyl residues, either intrinsically within the protein or with a binding partner. And so all the sapiens should work in this unifying method. Now, now we've talked a bit about lipid droplet formation. I'd like to take kind of the next frontier of lipid droplet biology, which is basically what happens at later stages. And basically, as I said, we kind of knew that sapien sits as ER-LD interface. It was already shown in 2013. But here, this is a new tomograph from Julia Machamed's group at EMBL, where they really can pinpoint directly sapien right at this interface between lipid droplet and this ER tubule here. And what's really going on here is that prior to lipid droplet formation, sapien will sit in the ER bilayer. Here you have the bilayer, and sapien will sit in with its luminal domain. And then when lipid droplets form, you get, again, this monolayer structure forming, but sapien is still sitting at this position. But here, it has NNC termini that have to be in the cytosol. So the transferrin domains have to open up in order to support this formation and traverse the bilayer. And so basically, this is something that's been thought of in the field, but no one's really shown this ever. Now, the question is, does it change conformation? This is what we suspect. And can we capture these membrane dynamics? And the short answer to this is we can. And this is actually Xenopus sapien. So again, another sapien, another beautiful ring. And we could actually capture a closed structure and an open one over here. And just to kind of get you on track of what's actually going on here, if we just look at this movie over here, this morph, you can see that the only thing that's changing here are these small linker regions between the transmembrane regions and the luminal domain. And actually, I can tell you that in yeast and in human and in Xenopus, these linker regions are really, really important for lipid droplet formation. So again, any sapin you will choose, they all need these linker regions to be flexible and to be able to open up. And if we go back here to this model I showed you, what we really think, what we've kind of managed to study is really how sapin promotes lipid droplet formation across the board. So we know now how sapin functions at the early stages and how it promotes lipid droplet formation, and now we're starting to understand how it actually stabilizes lipid droplet and what it does at later stages. And with this, I'd like to end and really thank, I guess, the two fellowships of the Sapin Ring. The first study was done with this group, with Susan and Justin, who are also in the second fellowship, and Robin Corey on MD Simulations, and Pedro took this. And really thank Pedro and Mike Wren as well. Mike Wren was a postdoc in the lab who helped me on this. And now thank Julia and Veo and Stefan and Jenny for their nice new collaboration, the other Covile lab members, and of course, all our funding. Thank you very much.
Thank you so much, Yoel, for your inspiring talk. Please questions for Yoel. If there is no question, I can ask one question. So does sapin play a role after the lipid droplets are formed?
Yeah, so that's a good question. We think it does in a sense. I just showed that it kind of stabilizes, but we think even once the drops are formed might even be important for the consumption. So I didn't talk about that we would need to consume energy, so we'd have to consume this lipid droplet. We think that's our kind of model would be the same as like a valve, and it kind of helps direct the flow of neutral lipids in and out, and kind of blocks as well from them from escaping. So that's kind of really the next frontier and what we're trying to address now.
Thank you, Yoel. We have time for more questions. But if there are no questions in the chat, I can ask the next one. So are there other proteins that promotes lipid droplet formation and homeostasis?
Yeah, so there are quite a few. We don't really know what they do. We have ideas about how they function. It's interesting in yeast there are quite a lot of them, and in humans it's thought to be a bit more simpler, which is strange. Usually not the same way. And I think that's something the field has to address now. What are other proteins doing, and how are they related to disease states? So that is also one of the next things that we need to start looking at.
Very interesting. Also,
I have a question actually, if I may, maybe a naive question, but do these lipid droplets, they bud off the ER, and can they be secreted?
So that's a great question. So in yeast, no. In human, people thought that they do, but every cryo-ET every lipid droplet has a neck, but they can be secreted. So actually, lipid droplets are secreted in breast milk. That's very well known, but that's like a very unique stage. So the field is a bit torn between. They always thought that lipid droplets in human cells can detach and even reattach. There are beautiful ex vivo kind of studies where they can attach and reattach lipid droplets, but every time we look in a cell, they always have that stalk connecting them. So it's a good question.
I have a general question, another one.
Yeah, go for
Talking
it.
about like a lipid droplet secretion, do you also look at targets for pharmaceuticals for lipid secretion, like obesity or other general obesity depending diseases?
Yeah, so lipid droplets aren't really they're not secreted in the sense. I know what you're thinking about. You're thinking about, when people so lipid droplets basically will be aberrant in shape, also in obese patients and in diabetic patients. So actually there's something going on there, but they're not really part of that secretion mechanism directly. So of course they store the energy, but they aren't like snipped off and then secreted, if that would be like in the model that I would think of. But definitely lipid droplets are important for this. We're not sure how. and we're not sure why they change shape, for example, in obese patients or in fatty liver disease and so on, and which proteins are governing them and what happens. So this is stuff that people now are looking at in more disease states. We were kind of in the field really torn about what Sapin is doing. Really, this has been like a decade of work on Sapin. Only the past 4 or 5 years, we finally figured out, our lab and some others. And now, Sapin aside, we can look at other stuff. So that's really the next stage.
Cool. Thank you so much, Yoel.